My name is Russ Harbaugh. I teach screenwriting in the School of Communications. And I'm grateful to have been a part of the Presidential Symposium Organizing Committee. And I welcome you to the 2025 Presidential Symposium, The Beauty of. I know a lot of you are freshmen, is that right? Or all across? I've been to two of the three panels so far today, and I'm feeling deeply nourished by them. And so I urge the students and my fellow colleagues, the faculty, to participate as much as you can this week. It is an enormous opportunity to connect with different branches of this university, which this is my 10th year, and I still feel like I can challenge myself every year to do more in that regard, and I'm always thankful when I do it. So this panel is presented by the Hofstra College of Liberal Arts and Sciences, School of Natural Sciences and Mathematics. They're presenting a panel on the beauty of seeing the unseen, scientific discovery from telescopes to microscopes. In this session, researchers from across the sciences will share snapshots of their work studying ordinarily unseen aspects of the natural world beneath our feet, inside our bodies, and across the cosmos. Please welcome our moderator for today's panel, Dr. Maureen Krauss, Professor and Chair, Department of Biology.
Welcome, everyone. I'm really excited about the panel we have for you today, and it does span the spectrum. We're going to start by the smallest, and we're going to go to the largest. So as we move, you're going to go from atoms and molecules all the way up to things you actually can see. And so the first speaker today is going to be Kevin Biscelli from the Department of Chemistry, and he will be talking about seeing previously unseen contaminants via high-resolution mass spectrometry. All right, so please welcome Dr. Biscelli.
Thank you very much, everyone. Can you, I don't think you can see my presentation, can you? Is there a secret? Maybe once we go to, oh, look at that, magical. Okay, wonderful. So yeah, thank you for the kind introduction. We are going from smallest to largest. So being in the chemistry department, I'm going to talk about atoms and molecules, which, as you know, you can't actually see. You're surrounded by chemicals. You're made of chemicals. We live in a chemical world. But we can't actually see them with visible light microscopes, at least. So I'm an environmental and analytical chemist. My research interest is in studying synthetic chemicals that we create once we're done with them. So where do they go? Do they end up in our food, our water? Do they pose any kind of a risk to us? And then what kinds of reactions might happen to them that would cause them to go away quickly or stay around for a long time? So very quickly, just talking about sort of the universe of chemicals. It's actually very hard to figure out how many chemicals humans have created. There's no single database where all this stuff goes. Every few years, somebody will get together, a group of scientists, and they'll come up with an estimate. 2015, the estimate was 150,000 new chemicals have been made. Chemicals and chemical mixtures, whoa, more recently than that. 2020, it was 350,000, so that's the number I'm going with here. And that was in 2020, so it's 2025 now. We're probably due for another one of these assessments. The key thing, though, is we have a tremendous number of chemicals around us. The vast majority of them are harmless. Many are probably quite beneficial to us. But we don't know very much about the wide variety of them. There's only about 15,000 that have undergone any kind of meaningful risk assessment. 120,000 of those 350,000 chemicals we know very little about. Sometimes we know almost nothing about them. They're just listed as a chemical that exists. There's a trade name. The chemical itself is confidential. And so, as I said, I'm interested in questions about whether these are causes for concern, what happens to them when we're done with them, what reactions they might undergo, and measurements are required for everything. I'm trying to demonstrate with this iceberg here how little we actually know about chemical space. So it's worth taking a minute to talk about how we measure these chemicals, at least in my lab. And so we use two techniques fundamentally. One is called chromatography, and the other is called mass spectrometry. So environmental samples, as you can imagine, have a lot of stuff in them, thousands of chemicals. And chromatography is a way of separating chemicals so that we can look at one thing at a time. you might have a mixture of chemicals. I've got 3 here. I put them into my chromatography system, and they come out as individual peaks. And then the mass spectrometry allows us to measure them. Specifically, it lets us measure their mass, and sometimes we can get insight into chemical structure. Sometimes people will talk about this as a fingerprint for a chemical, although it depends very much what kind of mass spectrometer you're using. So making environmental measurements are really challenging. For one thing, what you see depends on what you look for. So traditionally, you actually can't even measure a chemical unless you have an authentic standard that you can compare against. You actually need to kind of have a good sense of what that chemical is and that it's going to be there in the sample. It's very much unlike TV science, where you give me a sample, I put it in my magic instrument, and I give you everything that would be there. That's traditionally not been possible. We need a pure sample that we have to be drawing off of, and that limits our analysis really to previously well-studied chemicals, and it makes us blind to everything else that might also be present. Another major challenge is sensitivity of instruments. Environmental concentrations are often extremely low, and even though they're very low, sometimes they can still have meaningful impacts for ecology and public health. A typical drinking water limit for a pollutant might be 10 parts per billion. Parts per billion is like a pinch of salt in like 10 tons of potato chips. That would be like, you know, a giant semi-tractor trailer truck of potato chips. Or maybe it'd be like a scavenger hunt where I give a little postage stamp that's like a square inch, and I tell you it's somewhere on Hofstra's campus. You go find it. That's a part per billion. There's some chemicals, like you might be hearing about these perfluorinated chemicals, these forever chemicals that are all over the place and being regulated. Those are actually toxic at, or they have negative impacts in parts per trillion levels. So a part per trillion is like a grain of sugar in like 10 million gallons of water. It would be like that same postage stamp, except in all of New York, all of Nassau County, maybe. So these are traditional challenges we deal with. And just as a quick example of how new technology creates new opportunities, one of the beginnings of sort of the environmental movement was the creation of the electron capture detector. I'm showing you a picture of it right here. It was created in 1957, and it was able to measure chemicals that contain chlorine at very, very low concentrations, these part per billion concentrations. Once it was created, people started using it out in the environment, and it turns out that they found that there were chlorine-containing chemicals everywhere. They found this class of chemicals called CFCs, chlorofluorocarbons, that were ubiquitous in the atmosphere, and then eventually they realized that those were creating a hole in the ozone layer. They found that a lot of synthetic chemicals that were released as pesticides or for industrial purposes contained chlorine, and those were making their way into the far reaches of the Arctic, where they'd never been used before. This was a lot of the science underpinning of the sort of environmental movement, with Rachel Carson and Silent Spring. I want to talk about a breakthrough, if we can use that word, that is relevant to mass spectrometry, and that's high-resolution mass spectrometry. So as I said, traditionally, what you see depends on what you look for, and you kind of need to know a chemical is there in order to actually be able to measure it. High-resolution mass spectrometry really moves us beyond that in a lot of important ways. For one thing, mass spectrometry measures the mass of a chemical. Traditional mass spectrometry can do that to what we would call 1 unit. A lot of you guys, I think, are chemistry majors. I see some of you even in my lab. That would be like an AMU or a Dalton. That's enough to be able to say something there and quantify it, but it's not enough to identify something if you don't already have a reference. High-resolution mass spectrometry can go down to the thousands of a unit, and that actually is enough where we can actually pick up a signature of an unknown and actually be able to tell you the chemical formula of that unknown. And so that's a real breakthrough to actually get to that TV magic moment where you can actually start to pick up true unknowns, do what we call non-targeted analysis. The other thing that is relevant for high-resolution mass spectrometry is related to sensitivity. So traditional mass spectrometry can get to that part per billion level that you need to, but only for a small number of chemicals at a time. High-resolution mass spectrometers built fundamentally differently. They can actually measure simultaneously for all signatures that it might pick up, all chemicals that it's capable of measuring. It can go so low. So it actually can, in a sense, start to see everything, where you can actually use a magic magnifying glass and get structures. I think to 2021, we got one of these high-resolution mass spectrometers at Hofstra. They are about the price of a 2-bedroom condo. We got a grant from the Science Foundation, National Science Foundation. And so I'm going to talk to you about some of the work that we've been doing with it here. if I don't run over. So one thing that we're really interested in is what kinds of transformations happen to chemicals. And we're focusing on a particular chemical called 6-PPD. So 6-PPD, it turns out, is especially toxic to fish. The chemical itself isn't so toxic, but in the environment, it creates this thing called 6-PPD quinone that is super, super, super toxic. It causes extreme mortality after rain events for salmon and a lot of other fish, and it's toxic at concentrations at like 100 parts per trillion, so super, super low. And actually, the discovery of this chemical was one of the major high-profile breakthroughs of this high-resolution mass spectrometry thing. This chemical, it comes from tires. That's the common way it gets introduced. It's added to rubber to prevent the rubber from cracking and breaking. It reacts with ozone. And when it reacts with ozone, it creates this chemical here, this quinone. And that quinone is super toxic. Nobody knew that this PPD chemical was something we should be worried about. This quinone, there wasn't in any chemical database that exists. It was a true unknown that nobody had ever found. And it turns out that it's present in huge amounts in the environment. It might be as high as 90 million kilograms per year, because everybody's driving. Tires are as much as 2% made of this chemical. So huge amounts get introduced into the environment. This is not presenting the way that it looks on my slides, but I'm going to escape out of this for a second and kill this, even though we're trying to get rid of things here. What we're doing is we're interested in how this chemical is reacting with sunlight. So we're interested in whether or not sunlight can cause PPD and PPD quinone to transform. We're also interested in what kinds of things it might form. And so we've done a bunch of studies where we found that this chemical transforms very quickly, which is nice. It's not an environmentally persistent chemical. Also, it doesn't form this quinone under under sunlight alone. It probably actually will form this under sunlight. The chemistry is rather complicated. But if you take the pure chemical in just sunlight, it doesn't form. So that's actually good. The question, though, is you can see here, it does very quickly degrade. What does it degrade into? And high-resolution mass spectrometry really helps us with this. One, we can measure at very low concentrations. Two, we can actually see, okay, this chemical goes away. I can buy PPD. Since it was discovered, now you can buy it commercially. But what does it turn into? That's a very hard thing to do at very low concentrations, these parts per billion concentrations. With this high-resolution mass spectrometry, we can do that. And so we're interested in, it turns out the PPD is not stable in sunlight. Is the quinone, the really toxic one, stable in sunlight? One of the things we're interested in is whether chlorine from road salt actually will cause chlorinated quinone products, because we think that those are probably going to be much more toxic and persistent, right? Chlorine, as I'd mentioned earlier, is a bad thing when it's on a chemical in terms of environmental. I'm going to skip over another application we have on this in the interest of time, and just end by saying that this high-resolution mass spectrometry stuff is not just for environmental science. It has a lot of applications in medicine, public health, biology, forensics, lots of other disciplines. Whenever you hear omics, that's a HRMS application. And also, there are definitely some hurdles. I said it's like a magic microscope or a magnifying glass. You can see what chemicals are there. What you might be able to see are formulas. Actually making sense of that is actually a really computer problem. You get very large gigabyte size files that are 3 or 4 dimensional in space, and really this is a problem that's rife for machine learning. So if anybody has an interest in that sort of thing, it's something that we're actually talking with people at Hofstra about. I just very quickly, acknowledgements, students that have done a lot of this work, colleagues from other departments in Hofstra, places that have funded this, including the NSF, project I didn't talk about, funded by the DEC, and of course Hofstra University. Thank you.
All right, sticking with the theme of chemistry, we're going to welcome Dr. Sabrina Sobel to the stage, and she will be talking about making order out of chaos, making patterns in nature. So welcome, Dr. Sobel.
So I'm very happy to be here with my periodic table shoes and my starry sky sweater. Okay, so here we go. So this goes back to some research that I did with a colleague who has now passed away, Dr. Harold Hastings, and a bunch of students from the past. I'm trying to give you a bigger overview of a very exciting way that we can think about how chemistry leads to things that we actually can see. So we're going to be looking at chemistry that defies convention, the BZ reaction. That should not be the beginning. There we go. Order out of chaos, making patterns in nature much, much better. So we have the BZ reaction, we have zebras, we have cheetahs, and we have a brain coral. And so the first thing I want to do is I want to show the greater connection, and so that's going to be a YouTube video. So I'm going to escape out of this and go to the YouTube video. And it's a Minute Earth video because as you've probably discovered, there's a lot of good content out there, and so this is one example. So I'm hoping that this will have sound with it. So let's cross our fingers and see if we have sound with this.
Nature exhibits a seemingly endless array of patterns, from the cow's spots to
we
the...
do not. Sound but no video. Can we fix that? Full screen? Yes, I did full screen. That's not a touch screen, unfortunately. Is it second screen only or extend?
Try sliding the YouTube screen up.
Oh, yay!
Don't know if we can get PowerPoint back.
We'll find out. Okay, so let's start back at the beginning with our Minute Earth video. Okay.
Nature exhibits a seemingly endless array of patterns, from the cow's spots to the surgeonfish's stripes to the cheetah spots and stripes. But Alan Turing, the guy who invented computers, thought that those patterns might not actually be all that different. To show what he meant, he came up with a simple set of mathematical rules that could give rise to all manner of patterns. The rules go something like this. Inside each theoretical organism, there are two substances called an activator and an inhibitor. The activator stimulates production of both substances, while the inhibitor slows production down. In other words, it's kind of like a predator-prey relationship. The more activator bunnies there are, the more new bunnies they make. But a bigger bunny population also means more food for the inhibitor foxes, which means more foxes, which leads to fewer bunnies, which leads to fewer foxes. Except that Turing's rules say that as the populations of predator and prey increase, their ranges expand. Only the foxes need more room, so their range expands faster than the bunny's range. Soon, the foxes dominate the surrounding areas, leaving the bunnies in the middle to keep multiplying. So what we end up with is a pool of activator surrounded by a moat of inhibitor. And wherever the activator is more abundant than the inhibitor, it triggers some kind of change, like the production of pigment, which in this case gives rise to a spot of color. The beauty of Turing's theory is that by adjusting the variables, like the spreading rate of the activator and inhibitor or the total area of the system, you can get all kinds of patterns. For example, if you start out with slightly more activator than inhibitor in a bunch of spots, you might end up with, well, a bunch of spots. Or if the system is just a narrow little strip, like a snake or a tail, you might end up with stripes. Or if the activator spreads a little faster, it might leak out and join up with other patches of escaping activator, creating a labyrinth pattern. What's more, Turing's rules could be used to simulate patterns that closely mimic the splotches on cows, the stripes on fish, the mosaic patterns on giraffes, and even the tentacles on hydras. But the fact that his mathematical rules worked on paper didn't prove that nature followed them. And now, several decades later, scientists are still trying to figure out whether some of the patterns and structures in nature arise from real-life activators and inhibitors. On the one hand, we've found some patterns that happen in a very un-Turing way, like the segments in a developing fruit fly, which come predetermined by the fly's genetic blueprint. But on the other hand, we've also come across some tantalizingly Turing-like systems. In developing mice, for example, a protein called sonic hedgehog inhibits another activator protein with a less awesome name, producing stripy ridges on the roof of the embryo's mouth. And in its nubby limb ends, 3 different proteins activate and inhibit tissue growth to generate the stripe-like appendages known as digits. But regardless of how perfectly or imperfectly Turing's theory describes what we see in the real world, the coolest thing about it may be that it inspired biologists to go looking for evidence of Turing's ideas in living creatures. So observations inspired a theory that inspired observations that are bringing us a little closer to understanding how the cheetah got its spots and its stripes.
Okay, and since we are already on this, let's go look at the other video so that we have it in our mind. So this is going to be the BZ reaction, the chemical reaction that causes patterns in Petri dishes that I studied. Nile Red has a really short, wonderful video that shows you it in action.
One of the weirdest chemical reactions I've ever seen. To get started with this, I'll get a dish, and I'll add a mixture of sulfuric acid and sodium bromate, followed by a solution of malonic acid and water. Then into this, I'll shoot in a solution of water and sodium bromide, and it almost immediately turns yellow. This is just a bit of bromine forming, and I'll have to mix it around and wait for it to disappear. Now I can add the final ingredient, which is some ferroin indicator. I also have to mix this around until it looks pretty even, and after that, I just have to wait. At first, not very much was happening, but then I started seeing some small little spots appearing. Even more of them then started popping up, and they were all getting bigger and bigger. What's going on here is called a Belousov-Zhabotinsky reaction, and it's still one of the weirdest reactions I've ever seen.
Really
Today.
cool. Okay, so now we want to get out of this and go back to our presentation now that I've set it up. Ooh, it's coming up too. Yay. Okay, so this is where we were with the BZ reaction. So that's Belousov-Zhabotinsky, the chemistry that defies convention. And why does it defy convention? Because it is an oscillating chemical reaction. And an oscillating chemical reaction is one that doesn't just go to completion. It bounces between states until it uses up some critical factor. And this is like our activators and inhibitors. So if we look at the chemistry of the BZ reaction, we see that there are three main processes. And we're using a system that has a lot of bromine in it in different states. So to begin with, in our first process, A, we have bromate reacting with bromide and acid to make bromous acid and hypobromous acid. And then that bromous acid also reacts with more bromide and more protons to make even more hypobromous acid. So you can see that it's a cascade making a whole lot of hypobromous acid. And that's tying into making the bunnies, because the bunnies here are analogously oxidizing iron 2 to iron 3. So process B is the oxidation of iron 2 called ferroin in the complex. And so in that case, then bromate and bromous acid are going to make a really reactive intermediate bromine dioxide. And that bromine dioxide oxidizes the ferroin, which was red, into the ferriin, which is blue, and makes more bromous acid that can feed back into the cycle again. So again, you have this amplification effect. And then you have the inhibitor come in, and in the BZ reaction, this is called resetting the clock. And this is reduction of the iron 3, which is the blue ferriin, and then the reproduction of bromide, so you can run process A. In that case, then the ferriin, which is the iron 3, is going to react with malonic acid and bromomalonic acid to remake bromide, and it makes it in a non-stoichiometric amount. The stoichiometry changes over the course of the reaction, and then reforms your iron 2 or ferroin, making your solution red again. And so I did a lot of research with Harold Hastings and students on this reaction. We're looking at, does this reaction behave differently under different acidity conditions, different, I thought you were, and other different conditions looking at it. And so we could find that in the overall dynamics, we had this pattern formation that we could model very well. And in this pattern formation, you can think of each of these waves as being the process A, process B, process C, occurring over time. So when you plot that, you want to plot that as if you're looking at one point and asking what is going to be the changes in concentrations of the three critical species that we need to track, which are bromous acid in dark blue, bromide in purple, and iron 3, because that's ferry in blue, that's one is in blue. And so this is the plot that we get if we look at it as a time course at one point as the wave goes by. And this is important because then we can also relate that to other oscillating systems. We have a lot of oscillating systems out there that we can then relate back to the kind of chemistry interactions we have here, even though the details of the chemistry are going to be different. So if you want to look at applications of that, this is a great article to look at. Obviously, I don't have time for that here. So a heart is an example of a driven oscillator. And so that means it's driven by the sinus node. This is possibly because heart cells have part muscle activity and part nerve activity, so that nerve signal from that sinus node is going to cause the contraction. The sinus node is at the bottom of the heart, and so that's why we have the contraction starting there. And so you can see when you plot that, you also get this idea of this oscillation of behavior. And it's especially evident if you look at ventricular pressure in blue, that really shows that oscillating behavior. So even though the electrocardiogram doesn't look particularly oscillating, it looks repeating, when you look at the chemistry that's occurring, it really is an oscillating system. Another example would be insulin production in the absence of meals. So this is real-world data. And so this is a human who was given a continuous glucose IV drip over hours. And so even in a continuous intake of glucose, you see that the insulin is going to oscillate. It's not a clean oscillation, but it is the insulin production is oscillating over time, and the glucose content is mirroring that oscillation because as your pancreas puts out insulin, it tells your cells to take up the glucose. So again, you have another idea of this oscillating system. We also have circadian rhythms. Circadian rhythms are mostly driven by cortisol and melatonin. And again, as melatonin rises, we tend to go to sleep. As cortisol rises, we tend to wake up again. And that is, again, an oscillating signaling system that is biochemistry. And so in conclusion, many systems have complex behavior, and you really have to think of kinetics versus thermodynamics. So when we think of kinetics, in this case, we're asking, what is it doing over time? And in this case, it's unique kinetics. It's like a pendulum. It's oscillating back and forth before it uses up its ingredients. Whereas if we think about thermodynamics in chemistry, we usually think of it goes from reactants to products. Nice, even system. But this is one where it oscillates a whole lot before it gets to its endpoint. And so we see that they have been successfully modeled. And so you see it in temporal patterns, in circadian rhythm, and heartbeats, and insulin glucose. You also see it in our spatial patterns, in the spots and stripes in the labyrinth. And we saw those spatial patterns in the BZ reaction. So this is super cool. You can actually think about, instead of chemistry just being one unidirectional, you can actually have it go through these cycles over and over again. And they can be either driven, or they can be time limited, or they can be space limited. And that's it. Thank you so much.
Thank you. That was a wonderful example of a phenomenon that crosses discipline boundaries, right? So our next speaker is Dr. Brett Bennington from the Geology and Environmental Sustainability Department, and he'll be talking about the beauty of petrographic thin sections. Hopefully, there we go. And you're rolling. There you go. All right. Yeah, well,
All right, thanks for coming out this afternoon. So I'm going to be talking about these things called petrographic thin sections. And for the uninitiated, petrography, if we parse the word, it comes from the Greek petros, meaning rock, and graphia, meaning writing or really description. And so petrography is the science of describing rocks based on their texture and mineral content. And geologists use petrography to classify rocks. And our rock classification is a way of sort of figuring out how did that rock form. And that's one of the cool things about geology is when you identify a rock, you're not just giving it a name, you're giving it a story. If you identify a rock as a granite, that tells you all kinds of things about how that rock came into being and the processes that made that rock. It basically unlocks the history and tells a story, which is why geologists study rocks. Every rock has a story to tell. So to do this, to figure out, what a rock is made out of and what it is, one of the tools we use is we make thin sections, and a thin section is a microscope slide made out of a thin sliver of rock, about 30 microns thick, which is about a third of the width of a human hair. And making thin sections is, and my students will attest to this, as much craft as science. You start out with a big chunk of rock and you cut it on a big saw, big diamond saw, into a smaller, kind of rectangular piece, and then you trim that down on another saw, and then we take that rectangular piece of rock and smooth one side of it flat, epoxy it to a frosted microscope slide, and then we have a couple of machines that we can use to trim most of the rest of the rock off the slide, and another machine that then lets us grind it down until it's maybe 100 microns thick. And then you have to finish it off by polishing it by hand on a glass plate with different size grits. And that's the crafty part, because if you just do it a couple of swipes too many, there's nothing left on the slide all of a sudden, and you got to go all the way back to the beginning and start again. So it takes some time to get that down. Once you've made your thin section, we have a special microscope that we use for observing thin sections. It's called a polarized light microscope. And what this microscope does is it reveals the optical properties that different mineral grains on the slide have, and that helps us identify what those minerals are. What's special about this microscope is that it contains two polarizing filters called nickel prisms, named after the guy that invented them back in the 1800s. There's one prism that is between the light source and the specimen, and there's a second prism that's between the specimen and your eyeball. And the light that goes through the first prism is polarized so that only light waves that have a particular orientation get through. Polarizing sunglasses do the same thing. They eliminate much of the light and just allow light coming, with a certain orientation to get through. That second prism is oriented at a 90-degree angle, so it blocks the remaining light that comes through. So if you look through the microscope without anything on the stage, you just see black. But when you put a thin section on there, the light passing through the minerals is rotated, and then it can make it through the second polarizer, and you see it. And then the stage on the microscope spins, so you can rotate the mineral grains through the light, and you will observe things like different colors as the light is refracted, and you will see certain mineral grains will go dark and then go light at certain angles, and you can use that information to identify different minerals. So here's an application of this. I was asked by the lead architect who is involved in a redesign of the courtyard behind the Brooklyn Children's Museum to help her and her team identify some of the big boulders that they were digging up. So the Brooklyn Children's Museum sits on top of the glacial moraine in Brooklyn, and these boulders are glacial erratics that were transported by the ice during the last ice age. And they want to incorporate the boulders into the final sort of garden path that goes down through the courtyard, and maybe have some educational signage about what are these rocks and where did they come from. So you can see in the picture here that a dug up boulder is a pretty ugly thing. It's all dirty and weathered, and it's really hard to kind of tell what it is. So we took a sledgehammer and broke off some pieces, brought the pieces back to the lab at Hofstra and sliced them up and made a thin section, or made thin sections out of them. And this is what a thin section from one of the boulders looks like under the microscope with just plain polarized light coming through the bottom, but without the upper polarizer on. And you can start to see that, you know, there's some definite, you can see individual mineral crystals intergrown together. You see that there seems to be two populations or three populations of crystals. Some are opaque, some are kind of darker and seem to stand out. They kind of, in the microscope, they look like they're sort of popping up, and the lighter crystals seem to be receding. So we can start to identify those minerals, but really it gets cool when you engage that upper polarizer, and that's where the light show begins. You know, as you spin this around on the stage, you see things like the elongate crystals appear to be stripy, and those are twin crystals that have grown together, and those crystal twins are very distinctive of feldspars, particularly plagioclase feldspars. The brownish grains kind of stay brownish looking no matter what their orientation is. They go extinct every 90 degrees. We can identify those as another mineral, a pyroxene mineral called augite. And the opaque minerals are hard to identify because opaque minerals are just opaque minerals, but most likely something like magnetite, right? And you know, watching spinning thin sections on the rotating stage while you're looking through the microscope, it's a bit like watching a load of tie-dye shirts spin in the dryer. It's very entertaining. This is another chunk of rock from a different boulder from the Brooklyn Children's Museum site. So we made a thin section. Looks kind of the same, a little bit finer grained. We made a thin section out of that. And here's a comparison between the first thin section on your left and the second thin section on the right. And you can see they look kind of similar, except the crystal sizes are different. And in fact, we have the same mix of minerals. You have plagioclase feldspar, the darker looking pyroxenes. The reason for the different crystal sizes, again, I told you that, you know, rock mineral composition and texture tells a story. Large crystals mean the magma, the magma that turned into that rock cooled more slowly. Small crystal sizes mean it cooled more quickly. The large crystal sizes, which those are, you know, about a millimeter in size or so, those are indicative of a type of rock called a diabase, and our local source of diabase are the Palisades Cliffs along the Hudson River. The smaller crystals, which are more almost microscopic, are typical of a basalt, and there are basalts exposed in northern New Jersey in the Watchung Ridges. So that tells us where those boulders came from. One boulder was picked up by the glacier from the Palisades, brought over to Brooklyn. The other boulder traveled a little bit farther from northern New Jersey. Right? So with the thin sections, we can tell the story of those rocks. A couple years ago, I decided to resurrect a course we have on the books on astrogeology. And to teach an astrogeology course, you need some meteorites. So I started studying meteorites. And does anybody want to hazard a guess as to where you find meteorites? eBay. You go on eBay. So I started buying meteorites on eBay, which is highly addictive and was hard to stop. The meteorites you buy on eBay are usually slices, but they're not thin sections, so I had to turn them into thin sections. So this is a type of meteorite called a eucrite, and if you look at the thin section, the plain polarized light on the left, it's called cross-Nichols polarized light on the right, you see something similar to the basalt in the diabase, and that's because this is essentially the same kind of rock. But this is a meteorite, and the type of meteorite it's classified as is a eucrite, and eucrites are thought to come from the asteroid Vesta, which is the second largest asteroid in the asteroid belt. Now, this is essentially a basalt. So the asteroid Vesta is partly made up of basalts. Basalts are igneous rocks that form when you have a mantle and a crust, and magma from the mantle erupts into the crust and forms lava flows. So this is a lava flow from an asteroid that was large enough and got hot enough when it formed that it differentiated, melted, formed a core, a mantle, and a crust. This is a thin section that I made a movie of many years ago when we borrowed Apollo moon rock specimens from NASA. And yeah, this will work. So I'm going to move ahead here, right about there. So this is plain polarized light. There's the cross nickels, right? So there's the tie-dye shirts in the dryer kind of thing. Same idea. That big crystal in the middle is a crystal of pyroxene. The elongate needle-like crystals are plagioclases. And then there are some smaller blocky crystals of olivine in there. This is another example of a basalt, this time from the moon. And it was the samples of basalt, or the moon rock samples that came back from the Apollo missions that showed that those dark areas you see when you look up at the moon at night are lava flows. All right, we didn't know that for sure until we had actual samples. And so it's kind of cool. All these thin sections are basically showing us the same geological process happening on the Earth, happening on an asteroid, and happening on the moon. And then just to kind of finish off with here, most meteorites are a type of meteorite called a stony meteorite or an ordinary chondrite. And ordinary chondrites don't look very exciting when you find them. They just look like sort of dull pieces of rock. But if you make a thin slice and shine light through it, what you see is that ordinary chondrites are made up of these little blobs of mineral, which are called chondrules. And let me just. So this is just a thin section I made from a sample from a meteorite called Abu Panu. Individual meteorite finds are given names or number designations. And what you see is that these very common chondritic meteorites are made up of little blobs of mineral called chondrules. And those little blobs of mineral are actually little tiny igneous rocks. They're made up of the same igneous minerals you find in igneous rocks on the Earth and the moon. They were once described by an early researcher who studied them as droplets of fiery rain because they melted at some point. And the cool thing about chondrites is they've been dated and they are as old as the solar system. So these are samples of rock from the origin of the solar system. But the fun thing is that our models for how the solar system formed don't explain where chondrules come from. You need an event that gets everything hot enough that it all melts before you have any planets. Or it's possible that these are the leftovers from planet formation. We just don't know yet. Regardless, though, chondrules are absolutely stunningly beautiful objects. Plain and polarized light. This chondrule is made mostly out of the mineral olivine, and you have the small round grains of olivine, and then you have these like elongated bars of olivine. We don't really understand why different chondrules have different textures and different mineral compositions. Different types of chondrules show up in the same together in the same meteorites. But they're just endlessly beautiful. And one last slide there, and I will tell you that if I could do it all over again, I would get a PhD in meteoritics. I mean, these things are absolutely fascinating. Thank you so much.
Those are really beautiful. All right, we're shifting over now to biology. And so we have Dr. Javier Izquierdo from the Department of Biology, and he's going to be talking about infinite beauty underfoot, soil microbes, and how they could save the planet.
afternoon, everybody. So we're switching to living things for a little bit. I'm a professor in the Department of Biology, and we are talking here about the microbes that I work with. So I am a microbiologist. Microbiologists do a lot of different things. They work on viruses, on fungi, on protists, on archaea, and many of us work on bacteria. So these are the bacteria that we're seeing here, and these are under a microscope, and I'm going to point. That's just one of them. And the size that we're talking about is 1 micron. So for anybody that knows the metric scale a little bit, a micron or a micrometer is a 1,000th of a millimeter. That's how big they are. This is what we're talking about in terms of size and in terms of tiny little things. And my role as a microbiologist, or my interest as a microbiologist, is the diversity of those microbes that are out there. We don't tend to think about them, and that's a little bit about the title of my talk, this infinite diversity underfoot. I like to bring, when I talk about soil microbiology, I like to bring a prop that is a tiny little sample of 1 gram of soil, which is about half a teaspoon of soil, so you can barely see it from where you're standing. I'll put it here in the light. Maybe you see it, maybe you don't. You have to squint very hard, but it's a tiny little sample of soil. And within that soil, there is a staggering number of organisms, just in that tiny little bit of soil, right? We're talking about between 1 and 10 billion cells, individual cells. And that's not 10 billion cells of the same organism. We're talking about somewhere between, depending on the soil, we're talking about 4,000 to 10,000 different species. Species that are as different as you and I are from a plant, or as you and I are from a fungus. So that's a staggering diversity of different organisms in just this much soil that I was showing you just a second ago. And the part that I'm interested in is not just how different these organisms are. Yes, that's very interesting. I could talk to you about that for a long time, but I'm interested in what is it that we can learn from these organisms that they can do, right? In terms of all that diversity, there's certain mechanisms and certain metabolic capabilities that these organisms have that make them very interesting and also very useful to us. So the two main goals of things that we do in my lab are looking at microbes that can do these. They can do many, many useful different things, and we can talk more about those later. But the things that we're interested in seeing them, in learning from them, is how we can get some of these organisms to fuel the world, or fuel the future, make the fuels of the future. So that includes biofuels and bioproducts that come from waste, and microbes are very good at doing that. And also feed the world. What is it that organisms can do to help plants grow in environments where they usually wouldn't be able to grow very well? So the stories I'm going to tell you today are mostly about what those organisms, what is it that those organisms do. But I'm going to do it with a lot of pictures so that we are sticking to the theme of appreciating their beauty, their physical beauty, while also appreciating their metabolic beauty and their usefulness, right? So here's one of the topics that we pursue in my lab is microbes that can degrade plant waste or plant material and turn it into biofuels or biochemicals, so bioplastics, for example, or precursors of bioplastics. And this is a sample of compost, which can be applied to soil, growing on cellulose. So this is a scanning electron microscopy picture from taken in our scanning electron microscope in the department. That's a facility that we have in the Department of Biology that is actually run by Dr. Williams, who will be talking about it and about some of his work with that facility in a little bit. And this is a picture of the microbes that we're able to grow on just cellulose. Cellulose makes the most of plant material and is an important, very recalcitrant component of plant material, very resistant to degradation. So most of the carbon is there in a plant, and that's what you want to liberate. That's what you want to break down if you want to transform it into something else, biofuels and biochemicals. And so let me see if I can point with this. So the background that looks like big chunks of material that doesn't look like cells at all, that's the cellulose that these organisms are degrading. And only organisms that should be growing on cellulose from compost are growing in here. And notice the broad diversity of organisms that we can see in here. Shapes of all different kinds. Some of them have these weird elongated shapes. Some of them have a little bit of a sporulation going on, or spores being formed at the very end. We have all kinds of organisms with also gooey material. You might notice a little of that gooey material being surrounding some of the cells and some of the cellulose. And that has a role in terms of helping them attach and helping them colonize that material. What we have found in these are what we call enrichment cultures or mixed cultures. We have been able to find organisms that do all kinds of cool things in there. It includes organisms that are able to destroy the plant material, the plant cell wall, the cellulose, but also other complex polymers that are very difficult to break down. But there's also organisms that can produce biofuels like bioethanol. There's organisms that can produce bioplastics in there. And there's also, of course, so much more that we're starting just to scratch the surface of what really is in there. One of our favorite organisms is, so notice that the change, now we're going from very diverse to kind of boring, but still very beautiful, right? One single organism, so this is an organism called Clostridium clariflavum, or C. clariflavum, that we work with in my lab. And here is Clostridium clariflavum growing on glucose, that just like you and I, these organisms love glucose and grow very fast on it. And notice that I want you to look at the cells and how bare they are. So they're very elongated, and part of the elongation is how quickly they're growing. Usually these organisms, when they're growing in nature, they're rods, but not long sausages as they're showing up in here, right? We're going to see a closer version of that in a second. But this is on a soluble sugar, like glucose, that allows them to grow this well. When they're growing on something like cellulose, they have to be attached to the surface. If the carbon and all the food is really just on that particle, that's where they're going to grow. So cellulose in here is shown in green. The cells, you can see the shape of the cells in orange, and then the vast nothingness of the rest of the tube where they were growing is shown in black. So when we use the scanning electron microscope to look at what's happening here, this is just with a regular microscope, but when we start using scanning electron microscope, we start seeing some of the structures in the microbe that actually allow them to attach to that material. So now you see that instead of being naked rods, right, they have all these globular structures on the surface of the cells that allow them to attach, and also allows them to eat the plant material much better than they would if they didn't have that. These globular structures, so you're probably seeing those, and I don't have to really point them out, but I think you see them very well on the surface of all the cells. When we have gone a little bit deeper and look at where those structures could be coming from, we have been able to see that there are these complex protein structures that attach to the cell. So the cell is going to be at the bottom in yellow, and it has all these proteins that now we're going to the molecular level, not the cell level, but now the molecular level, that serve as anchoring domains for them. So if you see all those octopus-looking structures that you see over there, those are little dock places or dockering domains for enzymes that are specialized in breaking down plant material. And they're all connected to each other. So these are structures that allow them to place all their enzymes that are able to degrade that material in one place outside of the cell, but also proteins, complexes, that allow them to anchor them to the cell as well. And they have this whole variety of them to make these structures that you end up seeing as these massive protein structures. We've been able to do that with the help of genome sequencing. So now we're going deeper into the molecules and looked at DNA, the genome or DNA sequencing of what is inside of these cells. So each one of these cells has a genome, usually a circular genome. In the case of this organism, genomes vary from one organism to another. But in the case of these particular organisms, is about 5 million nucleotides. That means 5 million A, T, Gs, and Cs. And that accounts for about a little bit over 4,000 genes. 72 of those are those specialized enzymes, different specialized enzymes that allow them to break down different plant materials, right? So they can customize those cellulosomes, those structures that I showed you before, to have the enzymes that they need to eat that plant material or different plant material from one place to the other. And we have sequenced a couple of those genomes. A lot of that work has been done by students in the lab that are listed down at the bottom of that page. So different story, switching from the genomes of these microbes. The other part of what we do in my lab has to do with microbes that help plants grow. This is a root from a beach grass on Long Island. This is also taken with the scanning electron microscopy facility that we have here in the Department of Biology. And you can see that even though I've disturbed this plant and this root as I took it from the beach and brought it here to take pictures of it, you can still see a number of organisms attached to the surface of the plant. A lot of those organisms are part of what we call the plant microbiome. They're there to help the plant or have an interaction with the plant in terms of what the plant needs to do. And we use not just microscopy, but we also use other methods like growing them on specific media, microbiological media in the lab, to be able to learn something about what those microbes do. So for example, this is a plate that we use in my lab to detect if the organism is able to sequester or remove iron from the environment. So these are some organisms that we took from those roots, and we've been able to determine that they, on top of that, on top of living on the root, they're able to, you see those halos, those yellow halos that are on the surrounding area. That means that they have sequestered some of the blue dyed or stained iron complexes that they were subjected to. So they've been able to sequester the iron as a nutrient for a plant. And that's something that might be handy for a plant when they're growing in a nutrient-deprived environment like the beach. Other plates allow us to see things a little bit different. So that this is a pure culture that we're seeing in the previous plate over here. This is definitely a mix of different organisms that are growing on this particular plate. But something you can see right away are these areas that are cleared in the plate. Like each of these organisms has dropped a little atomic bomb in there and has created these clearing zones in there. That's something that this organism is making that is preventing other organisms from growing in there. Those are antibiotics that these organisms are producing, and those antibiotics might also be something that is useful. So now you have seen two different kinds of products that these organisms are doing on a plate that allow you to say something about these organisms. We have been able to develop assays that allow us to see if the organism is making, if the microbe is making some sort of plant hormone that helps the plant grow. And that allows you to start sort of narrowing it down to a group of organisms that we can test on a plant, right? These are now organisms that are clearly doing something useful for the plant and that we can test a little bit further. That doesn't mean that they're not beautiful. Aside from being useful, when we look at these microbes on the scanning electron microscope, this is one of those antibiotic producers. It has an interesting shape that you haven't seen so far, a little bit of a filamentous shape, squiggly long rods that are shown in here. One of our plant hormone producing organisms is this one right here that makes this long chains of rods. It divides, but it also, the chains sort of remain attached to each other, almost like cables that are bound in a little bundle together. This is a little bit of a close-up of what that looks like. But it allows you to start seeing a sense of, okay, and is that something that this organism has that allows it to also colonize the surface of the microbe or attach to the surface of the microbe? And just to go back to this idea of what they can do to save the world, because of course with my title, I promised a lot for an 8 to 9 minute presentation, but the goal is still there, right? The idea is to, aside from appreciating their beauty, it's appreciating their usefulness. What is it that they can do for us and what we can learn from them? So a lot of the work that we're doing in the lab, for example, in the panel on the right, has to do with, now that we have learned what these organisms can do, we also test them on a plant and see if, for example, that this strain SJC102 that I'm showing on the right produces larger roots and larger plant material at the very top. These are now not beach grass, but we're testing this on wheat and also useful other crops that we would consume. Nobody in this room has eaten beach grass. I hope that you haven't, but some of you have consumed wheat. And we're also trying to see, as you can see from the top panel on the left, we're also trying to see how to help these plants grow in places where usually you wouldn't do agriculture, right? Places with poor soils that are not equipped to be just like our gardens, but where we're more and more with an increasing population, we will have to grow our food. And with that, I'll thank you all.
Thank you, Dr. Izquierdo. Okay, so moving on now to the Department of Psychology, we have Dr. Emily Barclay Levinson, who's going to be talking about the brain sees itself. So real change of gears. Let's get you swapped out.
I know what it says. It's good. Hi, thank you all so much. Because I am so extremely young and cool, I'm going to start my talk with a meme. And actually a series of memes that I think you will sort of see a common theme across these. And what all of these memes are sort of getting at is this idea that the brain is an organ, right? But it is also us in some fundamental way, right? That your sense of self, your ability to, introspect, right, to experience your own thoughts, your conscious awareness, your ability to think and learn and study everything, including the brain, comes from the networks of cells that make up the tissues that make up the organ of the brain itself. And this is, fun to think about in that kind of like stoned in your dorm room sort of way. But I think that these moves are actually getting at the idea that studying the brain can feel very profound. And can induce this sort of sense of awe at experiencing an essential component of ourselves that we typically can't see. Right? By definition, it is very hard to see one's own brain, with the possible exception of certain instances of brain surgery where one is awake. But hopefully, most of us are not experiencing that in our lifetimes. And so we have to look for kind of other ways to, as directly as possible, experience the brain and study the brain. Now, because, the study of neuroscience includes the study of, sort of complex human phenomena, neuroscientists have actually studied what is that experience of awe, of wonder. This is some colleagues from the Netherlands who did an experiment using MRI, magnetic resonance imaging, which we'll take a look at, and showing participants images that were kind of rated high in inducing awe as compared to control images that were pleasant but not awe-inducing. The images that they used were all beautiful natural vistas, little snippets from like the Planet Earth documentaries, right? We can all kind of agree, like, the majesty of a sunset is very awe-inducing. And what they found in looking at the activation within the brain during this process was increased activation in what's called the frontoparietal network. So the frontal lobe of the brain in the very front, the parietal lobe of the brain back here together. Those parts of the brain are involved in sustained attention, focus, concentration, deep engagement with material, and reduced activation in what's called the default mode network, which is the connections, actually, several structures within the brain that sort of turn on when your mind is wandering, when you're ruminating, when you're daydreaming, sort of thinking about the self, but in a kind of just like relaxed, mind-wandering sort of way. And so this suggests the idea that the experience of awe takes us outside of ourselves, right? And actually creates, this sense of deep engagement and deep focus. Now, like I said, these are beautiful natural vistas. Does this hold true with the experience of scientific awe in other settings? Like if I was scanning everybody's brains here while you're looking at microbes or while you're looking at images of the brain themselves, would I see the same network of activation? This is the place where it'd be really great if I could show you like my data on this. This isn't actually my area of like research scholarship. I don't have those data. I have anecdata that suggest, yes, I would expect that to be true. And somebody in the future could take this as your own area of research and run with it. But what I do have to show you are the different ways that we're able to kind of give people access into the study of one's own brain, like the visual experience of one's own brain, right? How can we experience our own brains and induce a little bit of that scientific awe, that scientific wonder in a classroom context? One of our tools that we have uses electroencephalogram, or EEG, which measures these kind of moment-to-moment changes in the activity within the brain, right? So like I said, brains are made up of neurons communicating with each other, right? When one neuron signals another, it induces changes in electrical activity in that what's that called, a postsynaptic neuron. And this is what EEG is, using small electrodes placed on the scalp that can pick up on and measure those changes in activity very dynamically. We're going to see some oscillations, right? So harken back just a couple talks ago. And as you watch these data, this is kind of like real-time-ish data, just sort of scrolling through, watch for sort of changes in the nature of these waveforms, right? These electrical activity waveforms. Well, they're going to get like a little kind of smaller and tighter and closer together. And then they'll kind of expand again and become kind of larger, more systematic, and more regular again. And in fact, what you're seeing, right, so there's some tighter ones, there's some bigger ones right there. What you're seeing are kind of real data from a participant in class. We collect these data, right, in class, where the participant was asked to do something incredibly simple, right, to open their eyes and then to close their eyes again, right? And exactly at those timestamps, you see this dramatic change in electrical activity, specifically in what's called the alpha wave, right? This relates to sort of relaxation in the brain, and less alpha in sort of the middle segment there, right? Because they're literally taking in more visual stimuli when the eyes are open, right? And that activity actually changes. What I love about EEG is you can do it right there in the classroom. Here's a couple of my students applying electrodes to each other, right? And that students can actually see this come in real time. But it's not the brain. It is a measurement derived from the brain. It's interesting. It's fun to be able to like actually watch it change, right? Some of our experiments, if you induce high levels of concentration or stress, you can induce a little bit of detectable visual change in that EEG signal. But it's not exactly the same thing as looking at your own brain. For that, we need a tool called MRI, or magnetic resonance imaging. This captures those really like beautiful, crisp, detailed structural images of the brain using magnets. So this is what you may have had an MRI for medical purposes, same structure as we use in research, right? It has a bore, a tube that you go into. And a powerful magnetic field is applied to whatever part we're imaging, like the brain. Within the tissues of the brain, there are protons, little charged particles, and if we apply that magnetic field, they all kind of line up along a magnetic pole, right? So all of our protons are aligned. Then we apply a radio pulse, right, an RF radio frequency pulse, and the protons rotate out of alignment. We turn the pulse off again, and they all kind of tip back to being back and aligned again. And the speed at which they rotate back is different based on the characteristics of different tissues. And the energy that is released as they're rotating back is detectable and converted into images that we then see as the really crisp, high contrast differences of the different structures in the brain. And so this lets us really crisply see differences among many kinds of tissues, actually, not just physical anatomical tissues. This is a series of images from an MR tech whose name is Andy Ellison. He has this fantastic blog, and he got a little bit of fame by imaging a bunch of different vegetables and fruits and flowers, right? So you're looking at an onion, a persimmon, and a pomegranate. And the way that MRI images work is we collect basically a snapshot, like a flip book, right? A series of two-dimensional images acquired very quickly and cycling through the brain or onion over and over and over again. And then we can kind of put that series of structures of images together like a little flip book. So that's why this kind of looks like it's bouncing back and forth through that image. This is exactly what we also do when we are looking at the human brain itself. So this is a series of those snapshots collected like side to side, back to front, and then from the base of the skull to the top of the head. So 3 different views of the same person's brain. And it's a very meaningful experience to get to see your own brain, such that we actually use it as an incentive for participants who are coming into the study. Like, we'll pay them for their research participation, but the thing that gets everyone to go is like, get a picture of your brain. Like, we send it home with you. You can put it on your fridge, and it does induce a very kind of like meaningful sense of self. In a very unique way, something that you don't normally get to see, but at the same time you can kind of recognize instantly is you. And it's one of my kind of great joys of sort of the pedagogy of neuroscience, is being able to give that to people. At the same time, the actual human brain, right, a physical human brain, isn't even captured by MRI. This is still an image. And the only way that we can really see human brains, and again, most of the time we're not seeing our own human brains, right? But to interact with a human brain is through anatomical donation. This is where I actually don't have any pictures of our own students interacting with brains, because our medical structure lab, there's a lot of privacy involved. Our students don't have their cell phones out while they're in lab, which is wonderful. But we have a really beautiful anatomical gift program through the medical school, and people will actually donate their brains that our students are able to learn from. And many of my students, we bring the neuroscience club, we bring the intro neuro classes, and students do report that it's a deeply meaningful, beautiful experience to literally hold a human brain in your hand. Some of it is just the kind of interesting part of like, oh, it's bigger, it's smaller, it's squishier, it's a different color. But there's also that sense of this was at one time a being, right? One sense of self is tied to this organ that is now just an organ that we're using to study. So those are 3 sort of neuropedagogical tools that we use to understand ourselves through the study of the brain. And I do want to kind of briefly acknowledge the sources that have supported all of this research, right? The med school's anatomical structure lab. My MRI research comes from the National Institute of Mental Health. And then Hofstra supports our EEG work in class. Thank you so much.
These are really amazing. All right, so the next speaker is actually with the Zucker School of Medicine, and it's Dr. Zainab Nasrallah, and she's going to be talking about the vagus nerve. Not all who wander are lost. And so let's bring that up. So here we go. Welcome, Dr. Nasrallah.
Hello, everybody. I'm most excited to share what I know about the vagus nerve with you all, because for the last 3 years it has consumed my life. And so it's going to be so much more fun to tell you guys about it than it was to actually do the work for this. So thank you for the introduction for the nervous system. Today we're going to talk about a specific part of the nervous system called the vagus nerve. And before we get into that nerve and why we've spent 3 years working on it, I wanted to back up a little bit and just talk about general functions of the nervous system. So we just heard Dr. Barclay-Levinson talk about its functions in being us, right? It's your personality, it's your decision-making center, it's your memories, it's your learning center. It's the part of you that feels your environment. It's the part of you that moves your body around. But it's also the part of you that takes care of things that you don't even think about. Your heart rate, your blood pressure, your digestive system, how much oxygen you have in your blood. All of that stuff is taken care of by these yellow stringy things that you see on the screen. And I like the image here, but I had to put this up of the actual human nervous system that's been dissected, just to say, having just worked on a little part of the nervous system for the past 3 years, that this must have taken forever to do. Okay, and so for the nervous system to do all of the things that it does, it has to be able to communicate within itself and with the other parts of the body. And it does so through electrical signals, and the individual cells in the nervous system that are doing the work are called neurons. And what I'm not young and cool, I guess, because I don't have a meme, but what I do have here is, if you look really carefully at the image here, you see the little red dots moving up and down, right? Those are kind of the directions that the electrical signals are taking. So if we're thinking about an electrical signal coming up from the fingertip, for example, to the brain, that's called a sensory signal, okay? That's the brain taking information in. If we're thinking about the brain making something else happen, then that's called a motor signal, and that's represented by the red dots kind of coming from the brain down the spinal cord and out through the nerves. So the nervous system uses electrical signals to communicate within itself and with the different parts of the body. And it does so through nerves. A lot of the nerves that we're seeing in this cartoon are called spinal nerves. These emerge directly from the spinal cord, but some of the nerves that we have emerge directly from the brain, and these are called cranial nerves. The vagus nerve, in specific, happens to be a cranial nerve. Here we're looking at the base of the brain in the human, and you can see 12 pairs to be exact, of cranial nerves emerging from the base of the brain, and the vagus nerve is this one right here, cranial nerve 10, and you'll see it's bilateral, meaning we have a right vagus nerve and a left vagus nerve. So you might be wondering, why is it called the vagus nerve, right? That sounds like a place out west. Vagus comes from the word vagabond, which means wanderer. So because this nerve wanders throughout the body, they decided let's call it the vagus. So here's an oversimplification of the pathway that the vagus nerve takes. So we have the brainstem over here, and we've got two long strands that emerge from either side of it. These two long strands represent the vagus nerve. It travels down the neck, into the chest, and all the way down into the abdomen. And you can see all of the branches that it has, right? It's connected to almost every organ system that we have in the body, which no other nerve really does. What's not shown in this cartoon is the jobs that the vagus nerve carries out, or its connections that it has in the head. Okay, so this nerve is responsible for parts of your sense of taste. It helps us swallow. In the neck, it also helps us with phonation or voice production. In the chest, it innervates the airway, so you can see the trachea here. It innervates the heart, so it's involved with, maintaining heart rate, blood pressure, regulating how much air goes into the lungs. Once it works its way down into the abdomen, you can see it's connected to the gastrointestinal system, so the stomach, the liver, the intestines, and so on. So because, again, it's so widely distributed, I would say that this is probably the most, the most connected nerve, in that it's connected to all of these different organ systems, hence its name. Now, we have learned that we can stimulate the vagus nerve, and for reasons we don't understand, it can treat several disorders that are otherwise resistant to treatment. So here's what's going on with the vagus nerve. We have learned that if we place a pulse generator, similar to a pacemaker for the heart, if we place it in the chest, and we place electrodes on the vagus nerve in the neck, and we stimulate that nerve, that, for, again, reasons we don't understand, has been a successful form of treatment for things like epilepsy, treatment-resistant depression. It's involved in rehabilitation for patients who've experienced stroke, and most recently has been used to treat rheumatoid arthritis. And I put a little asterisk here as a reminder to myself that Northwell just implanted the first vagus nerve stimulator to treat rheumatoid arthritis just in August. So you're getting, this is hot and fresh, and you're getting up-to-date information here. So it's, again, one treatment option for all of these disorders. It is also being explored in other disorders, like inflammatory bowel disease, bipolar disorder, Alzheimer's, Parkinson's, and my personal favorite, the one I'm most excited about, obesity. I'm going to sign up to be the first vagus nerve patient. Never mind. So, again, we don't know why it works, but it does. So its success rate has been so great that now social media has taken over the vagus nerve. So if you just Google vagus nerve, you'll get a lot of articles, and a lot of them point to, like I said, social media. So here's an article, this was from a couple years ago, about resetting your vagus nerve to feel better. All right? Here's one on boosting your immunity by activating your vagus nerve. This one's my favorite. An owner's guide to the vagus nerve. This is like your car manual, right? You have an owner's manual for your car, but now there's one for your vagus nerve. There's an article about nutrients to support it. Look at this, the vagus nerve activity and social sensitivity. Like, your social life now rides on the activity of your vagus nerve, right? So all of this to say that it's been extremely successful as a treatment option for several disorders, but it kind of gets oversimplified in the social media world. So if you ever encounter this, I don't think it's true. I don't think any of this is true. Vagus nerve icing, who knows? All right. Now, it's not a silver bullet. Yes, it's a treatment for several otherwise treatment-resistant disorders, but it does come with side effects, like all things. And some of these side effects are limiting. So this includes voice changes. Patients can report a hoarse voice, pain, especially in the throat, cough, shortness of breath, trouble swallowing, nausea, and vomiting. Okay? So, I mean, I said before that the vagus nerve is connected to tons of organ systems, right? If we start stimulating it, it might make sense that we're going to see effects that we didn't intend to have, all right? So the question that we've been trying to answer, and several groups across the US have been working on this, is can we stimulate the vagus nerve without stimulating everything it's connected to? So in other words, can we target organ-specific parts of this nerve and try to avoid some of these side effects? So to answer this question, a division of the NIH, or the National Institute of Health, developed the SPARK program, and one of the projects within that is REVA, or Reconstructing Vagal Anatomy, which is the project we've been working on. And one of the goals of this was to go back and revisit the anatomy of the vagus nerve and really question everything that we thought we knew about it. And we set out to create a map, a roadmap of the vagus nerve. What does its anatomy look like? And when I say anatomy, I'm talking gross anatomy. What can you see with your naked eye? And then also, what can we see on a microscopic level? So when we first started this, we thought it would be this simple. We have a nerve, and we're going to put little landmarks on where we think things are represented. But it's felt more like this. 3 years of this, I've lost my mind at this point. So what we've learned is that its anatomy is highly variable, and it's really not simple at all. So here's a little bit more of a more detailed representation of what we've been trying to do. So I told you guys the nervous system is made out of neurons, and this is a cross-section through the vagus nerve. So this is a cartoon. Imagine that you slice the vagus nerve and you're looking inside of it. And all of these little dots that you see represent neurons, which are really long, skinny cells that are connected at one end to the brain and at the other end to whatever your target tissue happens to be. Brain, stomach, not brain, excuse me, stomach, intestines, heart, and lungs, and so on. And in this system, in this highway, we've got signals going up to the brain and then signals coming down from the brain. Okay, that's the vagus nerve kind of represents the whole nervous system, in that you've got bidirectional information flowing. So one of the questions we wanted to answer is, can we look inside of the vagus nerve and decide or determine if these bundles of neurons are organ specific? For example, is that bundle dedicated to the heart? Is this bundle dedicated to the lungs? Does this one go to the stomach? Does that one go to the liver? And if that's the case, can we just stimulate a bundle? These are called fascicles. Can we just stimulate, for example, the ones going to the lungs and avoid all of the other side effects that might affect these other organ systems? So in order to answer this question, we have to dissect the vagus nerve from human cadavers at the medical school and run our samples through a few different steps. So first step, again, is anatomical dissection. We also did a little bit of ultrasound. This is not real. This is a painting. But that's where we started. Then, once we extract the vagus nerve, and at that point, my team and I have documented all of its branching, all of the variations that we've seen in it that we can document by just observing it. The next step is to take that vagus nerve and run it through a machine called a micro-CT. So you just saw some MRI scans, right? Another type of scan that we can do is a CT scan or a CAT scan. A micro-CT is like a CAT scan, but you can't fit your whole body in it. You can only fit 1 centimeter segments of the vagus nerve in it. And this is the machine that actually looks inside of the nerve and decides where are the different neurons. Next, those samples come back to Feinstein, the Feinstein Institutes for Medical Research, where we do some immunohistochemistry, staining some of our tissues, try to figure out if our neurons are sensory or motor, and so on. And then we put it all together to tell a bigger story. Oh, I'm sorry, trigger warning. If you're squeamish, close your eyes. This is one of the pictures of our anatomical dissections. Okay, so this is from one of our donors at the medical school. And what you're looking at here is a little bit of the left lung. There's the aorta, the biggest artery in the body. And right here, kind of running right on down in between them, is the left vagus nerve. And what's fascinating to me, and again, you wouldn't know this if you didn't kind of look at it in an atlas and then look at it in real life, is all of the branches that emerge from it. Lots of these branches going into the lungs, lots of them going back here to the esophagus and to the aorta and so on. Okay, so that's what we've been doing. Ultrasound, it has been a nice tool to show us the inside of the vagus nerve, but here's what micro-CT can do. Right, you can really see inside and take a look at what's going on. All right, and there's a little bit of microscopy there. So the lessons that we've learned so far, I don't have a lot of data to share just yet. We're still analyzing. But what we've seen so far, just from the anatomical standpoint, is that no two vagus nerves look alike. Not in the same person, your right and left vagus nerve don't look alike, and not in between people or among a group of people. So it's as unique, I would say, as fingerprints. So we've noticed that branching patterns vary, the branch numbers vary, and then the target tissues, or what the vagus nerve is actually connected to, also varies. And then finally, if anybody's out there and thinking about a career in anatomy, don't listen to the people who tell you we already know everything about the structure of the human body, because we don't. Okay, the study of human anatomy is not finished, and this was a little bit of evidence for that. This is the REVA team. These are our collaborators at Temple University and Feinstein, and this is our dissection team. I just want to give a shout out to our medical students and our faculty who helped with the project. Thank you.
Thank you so much for showing up. I've been told that we do not have time for our very last speaker, unless he wants to come up and give a 2-minute version. And 2-minute version. Here we go. We're going to do a 2-minute version. All right. He's up for the challenge. All right. So Dr. Jason Williams from the Department of Biology, and he's going to do a 2-minute version while we swap out. All right. Here you go.
So I'll give you the snippet, because I know I'm in between you and coughing. Parasitic isopods. And I took my assignment to heart of I had to convince you that they're beautiful, because they're beautiful to me. But for me, the unseen beauty is the unnamed aspect of them. So a lot of what I do is describing new species. That's not mine, but it's a beautiful one named after Vader, Darth Vader, of course. So I work on the parasitic isopods, not the ones you're probably more familiar with, like pill bugs. So mine, are they really beautiful? I hope to convince you. Just 2 examples. From shallow waters on the left there, a hermit crab species from the south of Africa, and a deep water species here, deepest water species of parasitic isopod known. So these things live inside the branchial chambers of these hosts, feed on the blood, sexually dimorphic, as you're seeing on the bottom there, very tiny males and much bigger females. So part of the beauty for me is figuring out the mystery of the unknown in the adults, but also their larval stages. So I don't have time for this, but the challenger went out and collected a host named Bellis for beauty. This host we have found from the Arabian Sea with a parasite. You see that swelling there? There's the parasite. Female living inside of it produces these larvae, these crazy larvae with external yolk sacs that we have no idea what it's for. Yes, it's yolk. Yes, it's to feed it. But there's 850 species in this group. There's only like 5 or 6 that produce a larvae like this, and no marine species do an external yolk sac on their posterior end like this. So we described it. It's got 3 points. So we described it after Poseidon. I cheated. That's Neptune, but you get the point. This yolk, why? So that, to me, that's the big beauty in this whole system, is that we don't know the answer to the big evolutionary why question of how did these develop in an evolutionary sense. What's the advantage of this yolk sac in this larval phase? It's extremely rare in this broad group. So new species, new features, and did I do it in 2 minutes? I don't know, but I did my best.
Trying to keep everybody to 8 or 9 minutes is a real challenge. So thank you for doing the 2-minute version. So thank you everyone for showing up for this panel. I really appreciate it. And thank you to all of the speakers today.